My name is Heather Failing. I am the Executive Director of Molecular Diagnostics at Clinical Reference Laboratory. I also serve as the High Complexity Lab Director for our company, and I have a PhD in molecular genetics. Clinical Reference Laboratory, we serve as the exclusive laboratory for GeneME, and so we receive samples from GeneME, and these are cheek swab samples. So once we receive those in our accessioning department, we barcode those samples. To protect our data, we have an encrypted, secure information system. And once those samples are accessioned into our system, they're then transferred to our molecular diagnostics laboratory. And then step 2 of the process is the DNA extraction process. The cheek swab samples are then mixed with a solution that allows us to strip the cells off of the cheek swab so that we can then extract the DNA from those cheek swabs. And step 3 of that process is the qualification of the DNA. Once we've extracted the DNA through a series of experiments on our automated robotics instrumentation, we then check the quality of that DNA by way of an instrument that measures DNA concentration and quantity. And this is actually a qualifier to then do the testing. So we have a strict set of threshold criteria that those samples must pass to ensure that the results are reliable. Step 4 of the process is the actual testing process. Once those results are obtained and the samples are deemed to pass, we then test them for the genetic variants. And at that point, what we're looking at is 12 different variants or SNPs that are present within the DNA. And most laboratories test just one strand of the DNA. However, CRL processes testing for both strands of the DNA. During the course of the reactions, we also run sample controls, and those controls must pass certain thresholds in order to qualify the run and to make sure that the data obtained is completely accurate. And the last step, step 5, is the DNA analysis. The data is independently analyzed by 2 separate scientists, one of whom is a PhD in our laboratory. So that ensures another level of quality control before those results are then sent on to GeneME by way of our electronic data reporting system. We have an encrypted, secure information system that allows us to transfer the genetic results from CRL to GeneME in the utmost secure manner.